Hello, this is Wesley again with Stangls, and we're going to go into a bit more detail about what I do here. So we're going to go through my equipment. We're starting with the test tubes I use. They're 15 milliliter test tubes, marked up to 13 because 15 would be the cap. There's the bag of them. Then we also use these pipettes, 5 milliliter, just basic laboratory pipettes. Nothing too special about them. There's the bag of them. The slides we use are 25 millimeter by 76 millimeter, 1 to 2 millimeter thickness. Just basic slide. We use cover slips, 20 millimeter by 20 millimeter. We can go up to larger sizes, but that would change our calculations within our spreadsheets. So we just try to keep it consistent with what size we use. We got our slide. We got a little sample here from our Johnson Su Bioreactor. So we grab our test tube, goes off the scale, get our scale on. We tear away the test tube, and then we do 1 gram of our soil, mix it up to get it even. Hopefully get this on the first try, because that'd be pretty cool. As it falls off. Not enough. Hopefully get it on 2 tries, because that'd be cool. Not enough. And 3rd try is correct, as we chuck it again. Get that out of the way before we spill it. We'll take our pipette, grab some of our water, and we fill it up to 5 millimeter mark. That makes it a 1 to 5 dilution. Put the lid on and give 30 gentle shakes. I like that voice. Then we take 1 drop, which is 1/20th of a mil usually. That's what Dr. Elaine has told us to use. And we got our cover slips. And we slip that bad boy on, hopefully not getting too much sediment or air bubbles in it. And apparently I've taken a very bad sample. So, very bad sample. We're just going to redo that. So, same. So after 30 gentle shakes, grab our dropper, shoot it in and out a couple times to get a good sample. Not too much sediment. We drip it a few times back into the test tube to let any of the heavier sediment that came into the dropper get out easy. We throw a drop on there. That should be 1/20th of a mil, which Dr. Elaine has told us to do. Just take the cover slip. Try to clear out some sediment from there and get a good sample. Knock it a few times just to feel better. And that's pretty decent. Got a rather large air bubble in there, but that's fine. We can work around it. Pop over to the microscope. Get it down. And get that bad boy on there. Our microscope is an Omax model. We have 10x objectives for the eyepieces, and we got a 4, a 10, 20, and 40. So we multiply the 10 by these down here, so it's 40, 100, 200, and 400x magnification. Let's get this into view first. And we can see, not very well, but it'll do. So, what we're doing here is, imagine this is the cover slip. What we're gonna do is go back and forth just like that, all the way across the cover slip, viewing the entire thing completely, so as we can actually look for any larger larger organisms and nematodes as well, or worms, anything really that's big. Well, we just take a little peek and start going. Hopefully we'll get to see something. I mean, just based on the coloring, the green, and how dark it is, it shows that there's a lot of organic content within this. This is definitely a compost. Oh, we got a nematode. You can see him there, probably not. He's trying to hide a little bit under some aggregate there. He's moving pretty fast, so I don't know if we can focus on that. But basically all we can see is his tail. And usually we'd sit here and wait for him to poke his head out.
And right there.
Poke his head out to see what type he actually is. There's 4 types of nematode we'd look for: fungal, bacterial, root feeding, and predatory. And just based on his size and general shape of the tail, this could be a fungal or bacterial feeding, but doesn't look like he wants to be very cooperative with coming out. So we're just going to move on to keep things going for this presentation. Say bye-bye to that guy and keep going. There's that air bubble I was talking about. Very annoying to have in the slide, but happens sometimes. There's the air bubble. Oh. We got an arthropod. Wow. I guess he got caught in the air bubble a bit. I guess it is a good thing we had an air bubble then. Not sure if it's coming in very clearly for you guys, but he's pretty fuzzy. He's got 2 tails here, overlapping himself. He's probably pretty stuck just because of the air bubble.
Slide cover being on top of him.
Fighting pretty hard. Legs and everything. His head.
Look at that.
Antenna. He's actually pretty cool. Wish we could have actually seen him in the water, but caught him in the air bubble, so pretty neat to see.
What magnification are you on?
Right now we're at our 20x, so 200 times.
Go to 400.
Go 400.
Let's see. Oh,
show a few more details there. Actually focus on the tails. No, can't focus on the tails underneath him.
Yeah.
There's his legs and everything.
Poor guy. He's pretty well pinned.
Yeah. He ain't going anywhere, so I'm sure we'll see him again once we get back around. But let's just keep going. Usually we would mark down all these things we're seeing within our spreadsheet and keep track of it. But just for this demonstration, we're just going to skip that part. So we're just cruising around some more. Some types of seeds there. Get that focus. Here, here, and up here. Because of their size, there's a 4th and 5th one actually. Boom, boom. Can't really tell what they are. They're some kind of seed or a larger cyst. Well, really, that's just dormant life because it's too cold for them to survive right now. They'll become more active once it gets warmer and they'll become whatever they are and keep living. I'm getting pretty close to the edge now. It's getting pretty clear up. There's the edge. So that's basically what we do for a 1 to 5 dilution. Now usually what we focus on is we go all the way down to our 400 times magnification. That nice and focused. And the water is flowing that way, but if you can see it all these little moving pieces are all the bacteria in here. What we're supposed to do is count all of those, but considering how generally dirty it is and the number of bacteria there is, we're going to have to dilute further in order to be able to count those accurately. Well, let's just... Where'd Mr. Arthropod go? He's still there. Of course he is. Get on there, see if he dances around some more for you guys. Now for diluting further, grab our sample back. We're going to use the same dropper just because it's already contaminated with this, so it's not much point in switching. And we'll dilute all the way up to 10 milliliters now. Lid on, give that a shake. 30 again. Grab a single slide. Yep. And do a little wishy-washy again. And put a drop on there. One cover slip. And yeah, that's an alright sample. Usually we try to get that filled in, but got a lot of aggregate on the one side, so it's kind of lifting up the cover slip. And then we say goodbye to Mr. Arthropod here. Bye-bye. And we switch to the higher dilution. Even diluting higher, it's still pretty chunky, but hopefully we'll be able to do a count. I'll try to get to an edge right now, and we'll just do a quick zoom over at the, lowest magnification again, just to see if there's any real difference. Because we found the one nematode in this slide, but if there's like 20 in this one, we're gonna have to do more samples just to get an accurate representation. Just a little quicker zoom around. Nothing. There's our water's edge. So that's really it for the quick view. And then we go down with our magnification. Usually, yeah, I would dilute this another step just because of how chunky it still is and how numerous the bacteria are. Sometimes that's not always the best because diluting further means you're losing more and more viewing time, basically. It's like finding a needle in a haystack pretty well, kind of. The more hay you have, the longer it's going to take. But usually we do multiple samples, so even if we get a bad one like this one, we can still come out with the numbers that we need to. Let's just take a look around. And already right now, we've got some amoeba shell. There's an amoeba shell. There's an amoeba in his shell that's still there. That's another shell there. They're usually pretty active and more blobby moving around, but colder temperatures, they're trying to survive and stay dormant, so they're just hiding in their cells. If it got to even worse conditions for them, they would form into little cysts, protective barriers around themselves, just to really survive. Well, let's just zoom around a little bit, see what we can see. And there's plenty of amoebas and evidence of amoebas everywhere. We got a spore here. Numerous amoeba shells. There's a cyst like I was talking about. That could be any of the three protozoas, amoebas, flagellates, or ciliates. They all just form into pretty well the same thing when they're trying to survive the worst conditions they can. We've also got some spores around it, just little ones. That might be a spore. Yeah, that's a spore as well. I'm trying to find some fungal strands in all this mess. There's some small ones right here. Can't really get them to focus well. There's a big old crystal. We got quite a few spores right here. There's one here, 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 and up here as well. So yeah, this is a really good sample just because of the amount of green, the amount of spores, the amount of protozoa we're seeing. Yeah, that's basically what we do for that version, or that test, really. Usually, oh, I guess I should show that. Usually, oh, there you go, come on. Usually when we do the counting like this, if we imagine the cover slip again, we do 20 views. We would just start, say, in one spot, just randomly. Then we would jump to another spot, do a count in that location, randomly jump to another, just really going everywhere over the slide, not trying to focus on one spot, one good spot, one bad spot, trying to keep the views and the count partial, trying not to focus on anything. We would do that 20 times, take all those numbers, do a bunch of math, and come out with a fungus to bacteria ratio. So that would be the first method. Now, the second method we usually do as well involves using a hemocytometer slide. Glasses would help. That would be this guy. I'll show you with our, we have two hemocytometers here, a cheaper one and a more expensive one. The cheaper one was to learn to play around with it, really learn how to use it, but it didn't come good because the hemocytometer its little viewing area are these two little blocks. Inside those is a grid pattern that allows you to count the spores and whatnot. I'll explain further later. But the lines on these grids are all crooked, so it's not very helpful. But this guy has these channels. These sidebars are just a little bit higher than the viewing area. It allows for a measured depth, so we can get a better count and actually keep our math straight. Now, the proper one we use is this guy. I'm not going to be holding it up and waving it around like I just was, because I don't want to break it. But the depth on this guy is 0.1 millimeter. So that's our measured depth, so we can make a better count. The cover slips we use for that one is 22 by 26. It's an odd size just for the way it's shaped anyways. But for these guys, we use our larger tubes, 50 milliliter. There's the bag of them. And then we gotta turn our scale back on. A little tear of this guy. There we go. Grab our sample again. For this guy, we want to use 2 grams. Just because we're going to be diluting to a bit more of an extreme than we are with the other one. If we would stop trying to roll. There we go. Sample back. So that's about 2 grams of soil in our test tube. And then we grab our water. Shut the scale off and move it out of the way. For this guy, we dilute all the way up to the 50 milliliter mark. So we fill it most of the way with the water bottle. That's the wrong lid. And then we grab a different dropper so we don't contaminate our stuff. And we bring it evenly up to the 50 milliliter mark. And just for show. Then we do that, gentle 30 shakes. I went a little bit out of order here because we forgot to set up our slide first. Glass in here somewhere, so that's not good. Yeah. So for the hemocytometer, those 2 pillars on the side that I showed you earlier, we need to wet those down just a little bit. Okay. Because that will create it, because we put the cover slip on first. And that wetting down will create a good seal for when we add the sample to the slide. And then we do the little shimmy in and out again, grab our sample, drip out the heavy sediment. And then between those 2 pillars in the viewing area that I showed, we shoot our sample into there, both sides, letting it fill up underneath the cover slip. And this one you can't really do badly unless you overfill it, and then you got a mess on your hands and you got to clean it and dry everything, and just it's a hassle really. Slide that guy on. Glasses off. Because we also look for arthropods and nematodes and all that stuff, we'll be focusing in on those little channels that I showed you. These 2 little channels on the side where we put the sample into, just because those are deeper parts and that's where most of the sediment and any of the larger organisms will be washed too. So just to do our due diligence, I just like to quickly go through that, see if there's anything there.
Yep.
And mark it down. Yeah, you saw something?
Yep. Go up higher. You'll see it moving. So it was signaling.
Oh, yep, there it is. Guess I missed it. It can't always be perfect. Now we got a nematode there. Wiggling away here. I'm not going to be able to focus very well just because of the depth of the water we're looking at. But I can safely tell you it is fungal. You'll have to take my word on that just because, yeah, I can't get it very clear there. But I can get it clear up here. So well, that's a good thing to see, because that means there's plenty of fungal within the sample for the nematode to be feeding on. Go down that one trench. Go down these little side trenches for any overflow that comes out. The other trench. Focus. There's some nice root structure here. I like patterns, so usually when I see a root structure like this, it's kind of cool to see. Keep going. Back to the middle. That. Go across the middle. Sometimes there's an air bubble there. Can't do much about it. But majority of this is just root structure and other parts of the plant. Right here, if I can get it clear for you guys. Oh, that was my bad. A little bit too hard there. I'm not going to be able to find that again. I accidentally bumped it a little. So, and then we just go over the viewing area. See if there's anything in there as well. Nothing there. But right here, if I can get that clear for you guys, if you can see it, is the grid pattern that's etched onto these guys. Like I said with the other one, this one is a sticker on it, so it had all the grid patterns misaligned, but this one has it etched into the glass. So there's one on the top of the viewing area and one on the bottom, allowing us to do 2 tests on the same sample. So what we focus on are these corner squares with the larger corner areas with the larger squares. Zoom in a little bit to make it clearer. But with these larger squares here, so with those we can easily count the spores and whatever else is in there. So we zoom right in. Stay out a little bit for you guys. There is the first square we'd be counting. Something's floating across. No, that's just the flow. So this would be the first square we count. There's a total of 16 in each corner of the grid. So we would go through each of these squares and count the spores in them. For this one, we've got 1 spore, 2 spore, 3 spores, 4 spores, possibly 5. Yep, that is a spore there, so 5 for that one. Move to the next square. And Doesn't look like we have any spores in that square. Oh, we got one up here, so that'd be a total of 6 so far. Next square. Got one clear one in there. So 7. Hope I don't lose count because that'd be embarrassing. We got number 8. And number 9 on the edge here. Up here, here's one of the cysts I was talking about earlier. That's one of the protozoa just trying to survive. So I was at 8 or 9, right? 9? It's hard to keep count and talk at the same time. Usually I do it counting first, then talk. But we got 10, 11, and 12 here. 13, 14. And in that square, got a small one, 15 up here. Got a little bit of fungus coming out here. 16, 17. Got an amoeba shell here and a cyst. 18 spores, 19 spores. 20, 21. Small one, 22. Just knock it a little bit. I was at 24, correct? 22, 24, 25, 26. Another amoeba in there. 27, 28, 29, 30. All little ones. 32, 33, 34. 35. And we got 36, 37 in total. So we would add that total into our spreadsheet. We would do a count on each corner of the square, so we'd have 4 numbers in total. I'm not going to make you sit through the counting again. I know it took a while and it's probably boring. But usually we would add all those up, take the average of that, do some math as well, and we would come out with a total number of spores. Now if you wanted us to do, or if you were doing this at home and wanted us to do the math for you, all we would need would be that average number of spores you saw between the 4 corners and the dilution factor you use. We're using a 2 gram to 50 dilution factor, so that gets rounded down to 25, just to keep it simple. So if we did that, I'm not going to try to do it in my head, but 37 is a good amount of spores to see. Less is decent as long as you aren't getting below 10, 5 for the whole area. That it's still good to see some spores, but you want to see more. So really that's it. So since this is a Johnson Su, it's been going for 60 weeks as recommended. So we wouldn't see as much fungus mycelium, hyphae. So we would see more spores because it's in the process of reproducing. It's ready to go. It's ready to get out there and spread its seed. Just get this guy off the microscope so I don't screw it up. So that's all we really do for the testing. Usually we would do multiple tests on each sample just to get accuracy and really get back to you with accurate numbers and what we can do. Grab the sampler for me.
Go.
Okay, now for the samples we take, usually we would, as you saw, I only use 1 gram and 2 grams, so 3 gram in total. What we use are these little snack packs to keep the soil in. I would recommend grabbing sandwich-sized bags instead and give us a bit extra just in case I make mistakes or, problems arise, as they always do. But the sample taking, we have this T-sampler we use. It allows us to get into the soil pretty deep, I think about 12 inches this one is. But we really only need the first 2, 3, 4 inches. Now, as well, sometimes when I'm doing many samples, I'll also use this little guy. It's an apple core that I found at Lee Valley. It just allows me to jump it into the soil pretty good because it's spiked, and it comes apart so I can easily take the sample out and put it in the bag. Try not to handle the sample with your hands. That could contaminate it with whatever's on your hands, so it's always best to treat it as if you're picking up dog poop. So as well, if you wanted to send any of these samples to us, just contact us at the information provided. We'll happily do any testing for you. We'll get it done and get it done as accurately as we can. Now as well, I have this spreadsheet up on the screen behind me. We won't go into too many details because some of the calculations are proprietary, but the 37 spores I had within that one sample we looked at, it comes out to about 9.25 million spores, so that's a really good amount to have. So besides that, I will be putting up the presentation later. We've recorded the entire thing that we just looked at, so we'll be able to see better. We'll highlight what I saw in case you missed it with my finger pointing, so it'll just be easier. So if you have any questions, concerns, just contact us at the information. If I made any mistakes, let me know. It's always a learning experience. And I hope you enjoyed this and have a good day.